All right, well, thank you all for a wonderful session. While very inspiring, I think we also, this session really brought up all the details that need to be thoughtfully considered. Everyone else who's on who would like to unmute or be promoted to panelist to join in this discussion, we'd also appreciate it. So to start off the discussion and springboard off of a question that came from a patient in the Q&A session, I wondered if members of the panel might comment on some of the non-pharmacological options. Andy alluded to some of them in his talk with light stimulation, and Jeff had talked about those as well.
Well, I'm happy to expand. Andy mentioned that we've been piloting visual stimulation, and to do that, we hired a programmer to actually program into virtual reality goggles visual stimuli that we know from vision neuroscience happen to be particularly good at stimulating retinal ganglion cells to fire. And you might think, oh, I'm walking around my retinal, I'm sitting here right now, my retinal ganglion cells, aren't they firing a lot anyway? But it turns out that retinal ganglion cells respond most dramatically to changes and edges and motion, for example. And so actually, while we're sitting here, most of our peripheral vision is just looking at the wall and things aren't moving that much. So most of our retinal ganglion cells are fairly quiet at the moment. And so anyway, these goggles were programmed to provide very specific visual stimuli that we know are particularly good for different types of retinal ganglion cells. And I don't know if we've lost Andy here, but I don't see him on right now. But
he's giving a competing talk currly somewhere else.
Yeah.
But I did chat with him about Glenn Jeffrey's work, and Andy believes that it's relatively small in terms of the size. I think there was one question about the red light stimulation.
Yeah, and so that's another obviously totally separate technology idea. Anyway, so we're still like kind of in pilot mode for that. The visual stimulation leads to electrical stimulation of retinal ganglion cells. Another non-pharmacologic approach that's been studied a lot over the last couple of decades has been actual electrical stimulation of the retina. And so by placing electrodes outside of the eye, for example, on the skin on either side of the eye, and delivering small pulses of current, you can actually electrically stimulate retinal ganglion cells. And this has been explored with some, modest data, but there's still, suggesting that it could promote visual function in optic neuropathies. But there's actually really honestly a lot left to do in that. We're going to be starting to pilot some of these electrical stimulation devices as well over this year to come. And so I think there's a lot of excitement about non-pharmacologic approaches to promote ganglion cell survival and even sort of, regeneration or rejuvenation or, functional enhancement, let's just say, using these approaches.
Jeff, I wanted to ask, in your experiments with these new type of novel technologies, have you had preliminary discussions with the FDA about their acceptance of endpoints to show that these neuroregulatory therapies may be helpful?
Yeah, it's a great question. And actually, Len and I have had a lot of discussions over the last few years about endpoints, regulatory endpoints. And on the one hand, one could definitely design the trials to generate the data to give to the FDA around some of these newer biomarkers. On the other hand, as Len presented, the plausibility of using some of our traditional endpoints, including visual field testing in the clinic, the plausibility of designing trials around using standard endpoints that we already know the FDA is comfortable with as functional endpoints. It's so plausible now to run your trial in a reasonable fashion with those that I think it takes a little bit of the pressure off of thinking about, gosh, do we need a different biomarker for phase 3 trials? Len, do you want to expand on that?
Yeah, I agree with you 100%. It's not the nature of the intervention, I think, that makes the difference as much as the nature of the disease. And I think the creativity in exploring an endpoint, an outcome measure, comes from the disease. So the classic example, which the retina people on this session are well aware of, is the RP65 gene therapy trial, where there was really a lot of work done by Spark and colleagues to design the maze, which was really a luminance dependence navigation endpoint, which did reach acceptance. And I think we all know about in different diseases, some in ophthalmology, but mostly in neurology, where it's a matter of a conversation with FDA to essentially make the point: is it clinically meaningful? Will it actually make a difference in people's lives? And so for Drusen, why I maybe harped a little bit too much on the similarities to glaucoma, but Drusen, as all of us on the call who see Drusen patients all the time know, the classic, with the exception of developing an AON or a SRNVM, or occasionally a CRVO, it just looks like glaucoma. It's just occurring at a younger age in general. It's highly variable, and it doesn't, well, we don't know, but it doesn't seem to respond to intraocular pressure. People still progress very easily, even at low pressures in my hands. So why not just use, like Jeff said, the classic, endpoints that we already know? And if anything, our creativity should come in, given the nature of who we're treating, let's say the age of the patient, and for example, the nature of the speed of regression. We work with FDA to say, is this acceptable? But it still has to be clinically meaningful.
Talking about the treatment here, so certainly we try looking beyond pharmacologic agent, but in preparing for this presentation, I've seen a lot of data or case report on the use of pentoxifylline, which is Trental, a medication that's used for muscle pain, and we've seen a number of cases that optic disc drusen that have been treated with this. I certainly myself have not, but I'm wondering of the colleague in the line, Joyce or Heather or Len, have any of you ever used that for your patient?
There's some neuro-ophthalmologists who swear by it, but I think the majority of neuro-ophthalmologists are hesitant to prescribe that blanketly for ischemic optic neuropathy, for example, and certainly for optic disc drusen.
Okay.
Although I think it's a very interesting thing. I'm always someone who in my patient counseling has struck very strongly by what we have evidence for and what we don't have. But I remember at last year's Optic Disc Drusen Consortium meeting, the patients saying, that's great that you guys want a randomized controlled trial, but I'm actually willing to accept some risk for something that might help my vision. And so that actually caused me to think a little bit and change sort of what I tell patients and what I counsel them, and maybe not have this high level of filtering everything, but tell them what's out there. Because especially, there's so much on the internet these days. It's not really my exclusive knowledge anyways.
I'm curious, given that response, Heather, whether you, Joyce, Len, others in our audience, how you're addressing vitamin B3, which has received a fair amount of attention on the animal model side as being potentially neuroprotective, and of course is just available as an over-the-counter supplement, even though we don't have randomized trials in optic neuropathies yet.
To be honest, it hasn't come up. I give a lot of B2 for migraine.
I tend to give a generic advice of antioxidants. So the B's, the C's. I'm sorry, B's and C.
No E. So I think this is a great intro into talk about animal models, because I don't do the B3, partly because most of the data that was in the Science article was first published. Obviously it's all animal data, but there were some it wasn't as strong, I think, as to make me feel it's safe enough for patients. It was a single study. I know that you and I think the Columbia group are involved in a B3 study. I'm not sure what the results are. But I go by how good the animal data is when we have no quality human data. And if somebody really push comes to shove and somebody says, listen, is there anything you can try? Kind of like what Heather says, we don't really know. We're not gods. We're just ophthalmologists. We're like the lowest of the low.
God, like
We don't know.
neuro-ophthalmologist.
We don't really know what's the best thing to do. Then at least go by what's the best data. What are the best data? And the best data to me is alpha-2 agonists. You know, in terms of the animal studies, and at least one clinical trial, which was randomized, which was masked. It's just that they had a pretty high dropout rate. I'm talking about the low pressure glaucoma treatment study. So if somebody really, you know, says, you know, either we're going to shoot you if you don't tell me what you can do, or they don't say that, but it's something that, you know, what else can you do? That's the closest I can come. The trouble is often the people are asking are children, and, you know, there's always a risk with alpha-2 agonists in children. My strong belief, though, is that's why we really have to move forward in doing a proper trial. At least start with, you know, after the safety. You know, we have lots of choices here. You know, and the Stanford group, you are on top of the world right now and potentially doing a clinical trial. I don't think it's too early to start actually doing a plan.
We are part of the narrow trial, but we're very far from what Yasser and some of you have described. You know, I think it's definitely something that I'd be interested in, you know, talking to the people on this panel further about. We certainly have a lot of expertise and high interest in optic disc drusen right now.
And just to clarify, we haven't been involved in the clinical trials of B3, but yet we are trying to launch right now a randomized control trial. But B3, just to clarify for the audience, has been studied in high doses for a number of other diseases and found to be very safe and well tolerated. And then Johnny Crouston and the group in Melbourne actually published an ERG-based study of glaucoma patients last year, again demonstrating that it was safe and well tolerated, and that there was some improvement of the one of the measures on the electroretinogram. Anyway, so it's sort of you know, stimulating data, early days, obviously, but I think something worth exploring just because of the ease of accessibility for our patients.
So Jesse, you describe a lot of the endpoint, very high endpoint, and to that, Joy just described that's sort of like a very extensive, and maybe that's like a Lamborghini type of model. For us who just start right now with this, what are some of the easy endpoint that you think we can do? Like Joy said, we have a study now. How can we do it so that we can begin with a multi-center study, for example, but some of the endpoint that may be describe a system where it's a little bit less complicated than the one that you mentioned, or less detailed, because we may not be able to get there at this level yet.
Yeah, I think I should have included you know, another type of study, and that's the retrospective you know, studies, that imaging studies that where we pool data from various centers, and yes, it is cumbersome. They may have used different technologies and different types of imaging protocols, but we can potentially come up with a way to look at the, you know, the common, figure out what are the commonalities in the images that have been acquired, and maybe divide them into 2 or 3 different groups, and do sort of a pilot study where we could potentially come up with at least a strategy. Hey, all 5 centers have shown that different results for visual field, for example. And I'm not saying we shouldn't do visual field, but for me, it's easy to bash on visual field when compared to microperimetry. And we see that only 3 sites have done microperimetry, but they've shown similar results or similar trends. Then at least we can make a decision, okay, microperimetry is in, and we will try to figure out what's the best way to do visual field. The same is true for OCTs. Some may have used the swept source, some may have used the spectral domain. But if, for example, I'll give you an example of central subfield thickness. We have gone all the way from time domain to swept source, and now even, you know, multispectral imaging. The central subfield thickness hasn't changed. It is, you measure it with anything and it will be the same. It just depends on whether you include the RPE into your algorithm when you're measuring the thickness or not. So there are always opportunities for us to bring some sort of standardization to images that have been acquired through different devices. It's just, it requires some work, some legwork, and a bit of effort to pool that data. So it certainly can be done.
So we have a couple of interesting questions from some neuro-ophthalmologists in the audience who I'm trying to get promoted to panelist, and here they are. Eric, why don't you expand on your idea in general? So I won't mess it up reading it.
Oh, thank you. So yeah, I just noting the role of the other eye in regeneration that was brought up. I just thought that the sort of optimal approach may not necessarily be to remove stimuli from the other eye to gate suppression, but there has to be some other stimuli presented to the other eye that will optimally reduce suppression to promote recovery. I don't know what that is yet, but working on it. Maybe we'll figure it out soon.
If I could just say, Eric brings up an interesting point that actually was a point of study about 40 years ago at MIT, where you could take hamsters, and if you cut and let go of their optic nerve, nothing good happened. But if you had a crush model on one side and also remove the other optic nerve, you got some fibers that would grow back to the wrong superior colliculus, where the hamster is 100% crossing, which is sort of the old experiment of what you're saying. And I guess sort of amblyopia patching. So there's a trade-off between integrating the information from both eyes versus being able to have this, what was called the pruning effect, to permit the regrowth. And maybe even gets back to the biological reason that we don't have regeneration designed into our systems, because sometimes the integration is more problematic than losing an eye.
Just add one pitch on top of that observation. First of all, the visual stimulation experiment that Andy Huberman talked about and published also showed that blocking transmission from the other eye helped regeneration in the first eye. And I think it's also interesting to think about that from an activity perspective, particularly if the other eye's normal vision is just at a simple level leading to pupillary constriction, and removing it allows more light into the injured eye that you're studying. Could that increased access to lighter activity be one of the stimuli that's helping regeneration in that side?
Kathleen, did you want to expand on your proposal? If you want to unmute.
Oh, okay, I'm unmuted. And, well, this has been a wonderful conference, but I wondered about whether we should be thinking about a registry of optic disc drusen. There's a lot of questions that you can answer if you carefully phenotype optic disc drusen people from across the country and maybe even across the world, since there is the odds consortium. So I'm just going to throw that out, and your expert panelists in designing trials can comment about pluses and minuses of registries.
So it's interesting that you say that, and perhaps you were paid by the Nanos board. But one of the things that we've been working on is to explore the idea of registries more generally in neuro-ophthalmology, and how do you do that? And the odds consortium has been working on that and enjoying all the trials and tribulations of international contracts and data sharing and the like. I think registries offer a really exciting opportunity in terms of expanding the sample size, and also really truly getting at real world data, which tells us not only how diseases progress, but also what patients are willing to do in terms of coming in to get care and monitoring and things like that. I think the biggest problem with registries is the lack of standardized data, which just makes it hard to statistically compare at time points. And even if everybody has OCTs, if they're done by a different standard. Yasir was very nicely presented some examples of just the challenges of not only standardizing reading, but standardizing imaging. And so I think that would be a great idea. And we in the neuro-ophthalmology community are an enthusiastic bunch who I think could get together and sort this out and really lead to great advances in understanding these diseases.
And I certainly continue to be impressed by just the enthusiasm of the patients. Through the Optic Drusen Center, we hear from people all over the world, essentially, and it's I think they've inspired me to want to do more. And there are certainly a lot of challenges to doing a registry. Essentially, it requires some basic dedication from each site to streamline all these things. And perhaps since we have a lot of expertise in clinical trial design, and I love the presentation on the first natural history study and how that could benefit us. I think these are laying concrete steps forward, and certainly look forward to talking with Stefan Hammond and everybody on this panel into doing that, at least in some of the sites.
I think this is also, it's a great point to bring up, because just as we learned from trial design from glaucoma, we can also learn how some of those studies I showed you near the end on essentially showing that certain techniques are better than others all came from basically single center based registries, either at Duke or at Hopkins or at New York Eye and Ear. And they used, basically, most of them were based on fields. And they just, all that field data over many years, over many patients, was enough essentially to do all the kinds of modeling I just showed you as a kind of a Monte Carlo simulation. And I think as a very beginning, just simply, so simple, is just if every neuro-ophthalmologist who sees a Drusen patient, at least the ones who have access to a Humphrey machine, although it could be done with an Octopus too, ran a 24-2 C to standard and just kept those data and then got consent to be able to share them. That by itself, within 2 or 3 years, I think you'd have enough data to actually plan a trial. Forgetting about the OCT and all the other imaging, just the visual field, because that probably is the endpoint that you guys should care about.
I think the patients hit upon that today, that they were most concerned about their vision, not what we were seeing on the OCT, even though we were all excited about the pretty pictures on EDI. Yeah.
In
Sure.
terms of the quality of life, we have developed REDCap databases that can be piped in, and patients can be consented virtually and answer some questions. So that might be a good place to start, kind of like a grassroots organization, just to get the patient in, and then they could leverage their local neuro-ophthalmologist, and we could go from there.
Larry Froman put into the Q&A, we did try to set up, he's an ophthalmologist, we did try to set up registries at NANOS 10 to 12 years ago for Sussek syndrome and pre-MOG for autoimmune optic neuropathy. We set up standard forms to complete, and I think one person sent in the data. I certainly hope that this many years down the line, people are more willing, and I'm sure they are. I'm sure they are.
Well, we're about to try it again with venous sinus stenting, so stay tuned.
I think it's always come with nowadays, since many of us are so busy with many things. I think if there's some form of even some basic funding to help, because you would need a coordinator to at least help to keep up the site netting. Because I think if you truly just rely only on the clinician, unless we are so disciplined and so time available, it will be hard. So even if like a large center grant that just help to support a certain percentage of a coordinator at each site, where let's say Joyce or anyone can just say, okay, we have it here, can you help to put it in there? Otherwise it will be hard to that. So I think we should recognize the reality that it's very difficult. So we should try to see if we can help in any way.
Correct, and the more data we request, the more difficult it becomes.
Yes.
Patients are likely to be enthused to, log in, answer questions.
Yes.
They're probably also could likely go get a copy of their Humphrey visual field and upload that. But to start to get like the original files and everything else is going to require some provider and staff effort, which, as you mentioned, Juan, is all stretched thin as it is.
Yes.
You know, there's
Yeah.
a new law in California that I'm sure you're aware of, and I don't know if it's for the other states. It basically said from a month ago, patients can get all their medical records downloaded. I wonder if that couldn't be used to advantage in terms of patients working directly with the registry.
So it could be, but it turns out that it's the main medical record and that there's an exception for things that are in a different database. So they don't auto download, say, their ophthalmic imaging. Now we'll give it to them, and I know Joyce and I print it out for them all the time in clinic, but it's not, they can't go to a portal and just get their fields and things currently.
Well, my patients are getting all the notes, so maybe the visual field or radiology data is next, but yeah, right now that seems to be a barrier.
Alfredo, you mentioned, you had a question you put into the Q&A as well. That was a really good question. And maybe, I don't know if we want to read it or say it again to Jeff about the RFL thickening and target engagement.
Well, first of all, Jeff answered me privately, so
right.
I think that we all agree.
I think it's good for everybody.
The question was this, with neurotrophic factors, Jeff was able to demonstrate that the retinal nerve fiber layer got thicker. And that is very important because it proves that there was enough of the factor getting back there to be bioavailable to a significant level. But the fact that it got thicker, I think we all agree, does not mean that there were more axons there. It probably meant that the axons were swollen. And the fact that they got swollen could mean a variety of different things. More microtubules, more mitochondria, pathological swelling, or what would really be nice is the hypertrophy of axons that occurs with greater activity. And I probably left out another half dozen possible explanations.
Jeff, I don't know I don't know if Jeff's there right now, but he wrote, so true, we'll have to look for functional measures to complement structural ones. But I think this, I mean, to me, this is very unusual because we don't actually have any real good measures for showing in the human eye that if you give a drug topically, that you actually get a good effect at the retina. I mean, the closest we have is vitreous sampling from a drug that was done in a small number of our participants who are getting vitrectomies. Other than that, we don't have any standard ways of doing that, at least in the neuro-ophthalmology glaucoma fields. I know in retina, they did this, Genentech did a study looking at something, I believe that was in the aqueous or in the tears as a kind of a byproduct of the drug that they were studying. This has been published, I think it was in PNAS. But other than that, I don't really know of any evidence that you have a drug, you give it topically, it gets to where you want it to go.
Right. What I don't remember if we published this or not, but what Mark Kamayan and I had is this, he was doing this dog model of neurodegeneration. And in that, I was doing some pathology. And what was interesting is that the axons in that dog model were not dying, but they were getting skinny. And the presumption was that they were just not metabolically as active because they weren't being stimulated. Then when he put the electrodes in the area that the electrodes were sending an electrical signal, those axons got fat again. So that was sort of a restorative hypertrophy from stimulation, not to a drug, but to electrical stimulation, which is an interesting concept that healthy axons, or that the more axon works, the fatter it gets.
I'd like to throw out the question, and this may be sacrilegious given that this is the Optic Disc Drusen conference. But what we've discussed and some of the challenges with Drusen, of it being a rare disease, of us not knowing what the outcomes are, what are perhaps some of the best other models, either in animals or human diseases, that we might leverage to test some of these strategies?
Is there an animal model of optic disc Drusen?
I think Joyce is trying.
Not really. We're working on it.
Okay.
But the closest we got was, and Eric is familiar with this gene, the MFRP gene, which is known to cause familial optic disc drusen, as well as a nano-ophthalmos and a variety of pretty devastating eye issues. He took the mice, the MFRP mice, and looked to see if there are drusen there, but didn't find it. And you know, this is kind of related to what we were asking Tatiana Jacobs earlier about the fact that there's no mitochondrial calcification on the EM of all the animal models that she's looked at. I actually think that if we look for calcification or dysregulation of calcification, we may be more likely to find something, you know, as opposed to actually looking for mitochondrial calcification. So potentially the first step could be an axonopathy that involves calcium regulation and mitochondrial energy. So there's some candidate molecules that we're pursuing, but I think the biggest advance will be when we identify the first genes for idiopathic autosomal dominant optic dystrophy.
Your answer, Joyce, was so much more sophisticated than my thinking. But it did make me consider that question. Isn't it odd that in a disease that affects 2% of humans is not described in animals? And maybe it's one of the basic problems of animal models, because we don't usually look at animals that live for 75 or 80 years. And the percentage of people who have optic dystrophy at age 2, not so high probably. And that's the oldest mouse you're going to look at.
Although my mice are teenagers, so you know they could be having optic dystrophy if given the proper encouragement.
I see.
Can I ask a question, since we're roaming a little bit in a really good way? What about the nature of the vascular anomalies? You know, it just seems to be fairly common, which suggests that it's something that was developmental, and yet we, you know, it's clearly there's an association. I don't know how much of it's a risk factor. What might be going on?
Yeah, this was pointed out repeatedly in Mark So's 1981 paper, it's just unclear why. And he believed arteries and veins have this perivascular collection, and it doesn't seem to be calcified mitochondria, but just some kind of you know, cellular stuff or extracellular stuff. And when we actually just had a paper accepted today in Journal of Neuro-Ophthalmology, where we showed that multicolor imaging demonstrates this profound sheathing that surrounds the papillary vessels, same thing that we see in ischemic optic neuropathy. So there may be some important ways that we could interrogate the vasculature, maybe even give a little pentoxifillin, you know, some kind of stress test, and do a pre and post. I mean, I'm pretty sure we could achieve that, you know, just to understand sort of how tenuous the autoregulation really is in these eyes.
Well, apparently when you wear an N95 mask and a medical mask on top of it, you actually have a significant decrease in your OCTA measured vascular density. So maybe that's our stress test.
Oh, got it. Oh, that's fantastic.
Fantastic, but from a research perspective, maybe.
Yeah, we probably have to be a little bit more precise, but we can certainly slap on some kind of, you know, oxygen, you know, some kind of measurement like a smartwatch or a medical grade device to measure, just so we can standardize the levels while we do some vascular imaging. Good idea.
Getting back to Kwan's question, I think it was yours about the animal model. Whatever the animal model is, it has to be something that's a chronic one. I think, you know, we all in my own lab uses crush and, you know, acute ischemic optic neuropathy models all the time. But I think for this disease, one of the kind of hallmarks of it is it's really slowly progressive. And I feel that an animal model has to somehow take that into account. And there aren't that many slowly progressive animal models that aren't particularly selective. For example, there's small fiber models, but maybe those aren't so good because this is not necessarily a small fiber disease. In fact, if anything, it's the opposite. So, you know, I think there's a lot of limitation on what we could do as an animal model at the moment. I mean, you know, probably the most common one that people would use would be an increased intraocular pressure model.
So you remember, I was talking about the chronic issue too, that you want to look at old animals. And I remember that Brad Fortune had a habit every time that one of his monkeys died. These were monkeys to which a tremendous investment had been made. And I think they were macaque, and they would die at age 20 or 25. He would do histopathology. You might ask him if he's ever come across optic disc drusen. It wouldn't shock me if he didn't.
Yeah, the problem is these are probably animals where it could occur as part of a degenerative or some other process, whereas the human autosomal dominant optic disc drusen, the phenotype is just really striking, you know. There are multiple genes actually that are worth considering. For example, Pseudoxanthoma elasticum, which is the ABCC6 knockout. Mice have been around for a while, and that clearly causes vascular calcification as well as optic disc drusen formation. And there was a paper that was published recently that talks about the percentages of optic disc drusen being relatively high. I can't remember exactly, but something like 20%. You know, it's pretty high, much higher than the NFRP mutant. So, you know, I think they're good molecules. There's a list that I'm working off it, but there's nothing that beats actually identifying gene from the patients. And so I'm really looking forward to that.
All right, well, we're nearing our wrap-up time. Does anyone have any final comments on next steps or where to go from here?
Can we take a screenshot? We'd love to have a memory, at least on Zoom, of the people who are on this panel. Anyone who is willing to unmask themselves, go ahead and do that. We'll just hit the screen save.
While you're doing that, let me take this occasion to thank you, not only for this lovely conference, and I learned a great deal, but for having brought so many of my friends together on this common screenshot. It's more of a reunion than I had anticipated.
Yeah, I just have to also thank the neuro-ophthalmologists. I mean, there are a lot of polls, and you could tell, like, consistently the neuro-ophthalmologists are answering half the questions. So, okay, so we're gonna do screenshot smile. Thanks, everybody. So you're welcome to stay unmasked. I think Jeff and I are just gonna wrap up the meeting. Oh, wow, look at the time. We're exactly on time. That's amazing. Thank you, the tech support, for launching the last scientific poll questions.
Well, Joyce, I don't know if we've prepared for the finish here, but I'll just say it's been a wonderful day, a fantastic meeting. Very grateful for amazing tech support along the way, amazing lectures in all these sessions, great discussion as always. And so congratulations to you and the team for putting together an amazing meeting. I've had nothing but enjoyment all day.
It's been a fantastic meeting. I prepared just a small number of slides, mainly updating my thinking throughout the day. So anyone who have things to add, please do. Essentially, the idea, the hypothesis is still that there's either an axonopathy or some kind of event that leads to this optic disc drusen formation, enlargement, progressive calcification that messes the unmyelinated optic nerve, which then seems to fall in this autosomal dominant pattern. The vision loss, I feel that people have identified multiple areas where intervention can be done, including reducing the triggers. So even though we don't know exactly what causes the drusen to progress or for ischemia or edema to occur. We know enough about the optic nerve that these are pretty much a standard list. One of the things that hopefully we could get into more next year would be looking at novel biomarkers. Len mentioned the fast progressors. I would add to essentially markers of fast progressors. And we're looking at the patient's blood as well as skin biopsy to grow fibroblasts, where we could use seahorse or other methods, EM, to look at their mitochondrial function, as well as to grow induced pluripotent stem cells, generate neurons from them as a way to test the biology and the possibility of effective treatment. There's a whole field of ectopic calcification that I've been enjoying learning. It's actually not rare. And the reason why it occurs at the unmyelinated optic nerve head is obviously still a mystery. But I did come across some animal models. One of the most promising is actually, it's a spinal cord injury. It's a trauma model where the mitochondrial calcification occurs transiently and then disappears. So there's all kinds of cytoplasmic calcification as well as mitochondrial calcification. And so hopefully some of the research will bear fruit, and we'll bring you some of that next year. So I just wrote some of these goals while the panel 4 was discussing. We talked about early detection, identifying fast progressors or markers of progressors, optic Drusen genes, animal models, understanding the pathophysiology. Anthony Norcia and Jeff Goldberg were collaborating to look at functional changes using BEP. This is a 120 electrode system, trying to see to address Alfredo, your question about which population of retinal ganglion cells may be preferentially selectively lost in these patients. And then finally, I thoroughly enjoy session 4, looking at novel therapies. I thought Len and Quan, their emphasis on looking at axonal protection, Jeff and Andy's combination of regeneration, excitation, addressing things from the eye perspective as well as from the end target perspective. And then I think there's some very clear goalposts in terms of a first step natural history study or registry where we could actually come together more as a group in a rigorous way to identify the best imaging modalities or certainly protocols, in a way that would satisfy a eventual reading center clinical trial type. If we could, so Len is just an expert in this kind of power calculation, and I was actually quite thrilled by the numbers that Len got, and certainly hope to engage further with this entire community on making these things happen. So I want to thank all the speakers and of course the patients and their family and loved one, just in terms of contributing to this amazing meeting and for inspiring us to do more. I also want to thank the people who have been silently supporting us. We've been practicing and making sacrifices. So instead of doing experiments, basically they were prepping for this meeting. They've been texting you, telling you to get ready and sharing your talk and making sure everything goes well. So you really want to thank Brianna, Catherine, and everybody on this page. I've misappropriated members of my lab essentially to run this meeting. So thank you all. So I think we're now sharing the result of the last talk. And I want to mention that we're trying something different for this year. We thought it would be fun to set up a Zoom recording account in case people want to leave a audio or audio visual memento of this event. I, in particular, encourage the patients and their loved ones, because your words will, be inspirational to other scientists. A lot of people wouldn't know to study this disease if they didn't hear from you. And then for the scientists, just the camaraderie and then seeing everybody, I think it'd be kind of historic to have like Alfredo or Len's voice. We can listen to it in 10 years and think back, how far we've come, hopefully. So I'm going to leave the Zoom link in chat, and you will also receive it in the mail. So in case you want to share a little memento, basically as soon as you go in, it starts recording, and we will be extremely thoughtful in terms of the editing, and nothing will be shared on our website without your official permission. So thank you, everybody.
Thank you.
I guess I can't copy and paste too many things at the same time. Let me do it twice. Great. All right, well, thanks everyone. You are free to go. It's past 3. Thank you so much for all your time. We'll
Thanks,
see you.
Joyce.
Hopefully.
Thanks, everybody. Good to see you all.
Thanks so much, Tatiana.
Thank you very much. It was a great conference, really.
I'm going to email you
Thank you.
about staining for calcium in your sections.
Yeah, I'll have a look.
Okay.
Okay.
Take
All
care.
right.
Thanks, everybody.
Thank you.